SCFA and GPR41. Passage 1. Lipids and proteins are not only essential nutrients, but also act as signaling factors for various cellular processes. For instance, short-chain fatty acids, SCFAs, stimulate the G protein coupled receptors GPR41 and GPR43, whereas ketone bodies exert an antagonistic effect on GPR41. So, this introductory paragraph is essentially just giving us a little bit of background about how our lipids and proteins, our macromolecules that we know, are important for nutrients, can also act as signaling molecules. And then it gives an example of what the short chain fatty acids do by stimulating our G protein coupled receptors, GPR41 and 43. So I think it's important to note those are stimulated by SCFAs, our short chain fatty acids, and that ketone bodies exert an antagonistic effect on GPR41. So they're essentially inhibiting the function of GPR41 by antagonizing it. Researchers have long been interested in the effects of such metabolic intermediates on cardiac health. To test this, researchers generated mice lacking GPR41 and GPR43 and measured heart rate, a marker of sympathetic tone, in response to the SCFA propionate. I think these first two sentences are just kind of explaining our experimental process that we're generating mice lacking in GPR41 and 43. And then we're testing their heart rate, which shows us kind of like how their sympathetic tone is. And it's a measure of cardiac health. And it's important to know this is in response to the short chain fatty acid propionates. In addition to the results shown in Figure 1, the hearts of GPR41 mice, but not GPR43 mice, exhibited a reduction in the expression of tyrosine hydrolase. So this is some additional data that we don't see in Figure 1, and it could be important to the questions we're going to answer later. So I think it's really important to note that in the hearts of GPR41 only, mice that don't have GPR41 only, we saw a reduction in the expression of tyrosine hydroxylase, or TH. But we also didn't see that in GPR43, so that's important to note. TH catalyzes a conversion of L-tyrosine to L-dopa as a precursor to dopamine, a molecule which positively affects myocardial contractility by increasing electrical conductivity in the heart. And so this sentence is just explaining what TH, our tyrosine hydroxylase, does. And that is that it converts L-tyrosine, one of our amino acids, to L-dopa, which is a precursor to dopamine. And dopamine positively affects myocardial contractility, so how our heart cells are contracting, by increasing the electrical conductivity of the heart. So this is just important to know what TH is doing in case we get a question about it later. Figure 1, changes in sympathetic tone after administration of propionate. So on the left here, we have our y-axis, which is the delta heart rate, the change in heart rates in beats per minute. And on the y-axis, we have time in minutes after the administration of propionate. And I'm assuming this is the SCFA propionate because we saw that up here in the passage. And what's interesting, we see here that the wild type is represented by our white dots, and our GPR41 deletion mice are represented by the black rectangles, and our GPR43 minus mice, or GPR43 deletion mice, are represented by our black diamonds. And what's important to note about this figure is that we see actually a slight increase in the change in heart rate in the GPR43 mice, but not in the GPR41 mice. And so we see this change, and we can recognize that the changes between GPR41 and GPR43, and maybe that makes us think back to the expression of tyrosine hydrolase, which changed in GPR41 mice, but not in GPR43 mice. Researchers then wanted to clarify whether propionate acts through an adrenergic receptor, thereby promoting sympathetic activity, or a cholinergic receptor promoting parasympathetic activity. Um, so this is important because it's explaining the next steps in the researcher's experiment, and it's also explaining the two receptors they targeted and what those receptors are responsible. So I think it's important to highlight that the adrenergic receptor promotes sympathetic activity, our sympathetic nervous system, and the cholinergic receptor promotes parasympathetic activity, so our parasympathetic nervous system. After pre-treating mice with propionate, wild-type mice were given a dose of either hexamethylammonium, a cholinergic receptor antagonist, or propanolol, a beta-adrenergic receptor antagonist, which is structurally similar to our propionate that we were talking about earlier. So what's important in this sentence is that it explains the experimental design, that they're giving a cholinergic receptor antagonist or a beta adrenergic receptor antagonist in propanolol. Now we have figure 2, mice treated with drugs impacting the autonomic nervous system after pretreatment with propionate. I'm assuming this is the same propionate we used up here, the SCFA propionate. So on the left here in our figure, we have our heart rate in beats per minute, our change in heart rate in beats per minute. And on the x-axis on the bottom, we have the different experimental conditions they're exposed to. So all mice were exposed to the propionate. Our control mice did not receive either hexamethonium or propanolol. And we see that it has an 80 beats per minute. And then when treatment with only hexamethonium, which remember is our cholinergic receptor antagonist, we see no changes. But then when mice are treated only with propanolol after this pre-treatment with propionate, we see that the change in heart rate is drastically reduced. So this might be telling us that propanolol is important, and the because it is an antagonist, propanolol, the beta adrenergic receptor antagonist, is important to reducing the change in heart rate. So that's kind of showing us that the beta adrenergic receptor might be implicated in the way the propionate acts to raise heart rate, as we see in figure 1 with the wild type mice. Question 1. In the hearts of mice lacking GPR41, the concentration of which of the following is most likely increased? A. L-tyrosine, B. L-dopa, C. Dopamine, or D. Propionate. So what do we know about the GPR41 minus mice, the mice that lack GPR41? Well, we know that from the passage, we noted it earlier, that they exhibited a reduction in the expression of tyrosine hydrolase. So if we're reducing the amount of protein, tyrosine hydrolase, what is that going to cause? To find that out, we have to look at what the passage says about our tyrosine hydroxylase. So we know that TH catalyzes the conversion of L-tyrosine to L-dopa, the precursor to dopamine, a molecule which positively affects myocardial contractility by increasing electrical conductivity in the heart. So if we're reducing our expression in tyrosine hydrolase, we're not able to convert our L-tyrosine as much into L-dopa. So that's going to lead to an increase in our L-tyrosine. I'll just do LTYR here. Now, if we're not converting our L-tyrosine into L-dopa, we're going to actually have a decrease in L-dopa compared to wild type because we're not converting anymore into our L-dopa. Along that same line, if L-dopa is a precursor to dopamine, we're not going to have as much dopamine, and that gives us our answer. L-tyrosine, A, is the correct answer because it's increased because it's not being converted to L-dopa. So we have an increase, and then we have a decrease actually in L-dopa and dopamine because L-dopa is decreased and it's a precursor to dopamine. And then we can kind of get rid of propionate because there's not really a change in the propionate that's given to GPR 41 or 40 or GPR 43, and we don't really know. There's not really any mention of what's specifically different about mice lacking GPR 41 in respect to propionate. Question 2. Which amino acid is most likely found in the cytoplasmic domain of GPR 41? A. Alla or alanine. B. Met or methionine. C. Val or valine. And D. Ser or serine. Now, this question's really testing our knowledge of the amino acids and also what the cytoplasm is like. So, our cytoplasm is an aqueous environment, and that means it's going to be very polar and have charged ions in it as well. So, it's going to be um, very accepting of amino acids that have hydrogen bond donors, hydrogen bond acceptors, ions or charges, and it's not going to be very accepting of our hydrophobic amino acids. So we know that the cytoplasm doesn't like our hydrophobic amino acids, so it's likely that our hydrophobic amino acids are not going to be on our cytoplasmic domain. So alanine, that is a hydrophobic amino acid, so we can remove that from the running. Valine, also very hydrophobic, it's long carbon chains. Generally, we consider methionine to be hydrophobic because um, our cysteine, our thioether there is not going to be participating in any sort of hydrogen bonding and doesn't um, hold a charge whatsoever. And it's also accompanied by a long aliphatic chain on its side chain. And that leaves us with serine, which is the correct answer by process of elimination, but also because serine has an OH on it. And that OH can interact with the aqueous cytoplasmic environment because it has a hydrogen bond donor in its hydrogen atom on the OH and a hydrogen bond acceptor in its oxygen. And this causes it to be polar and interact favorably with the aqueous environment of the cytoplasm. So D, serine, is our correct answer.